Okay, good morning, everybody. Welcome. Happy Friday. So we got a lot of stuff in this chapter, some of which, as I mentioned before, really probably some review as we start jumping into some features of cells. So some of that we'll go through relatively quickly. Uh there is I believe it's a YouTube video that I linked up on here about Ignaz Semmelweis. We're not going to show that in here, uh, but I recommend you check it out. It's interesting. So when I think about old Ignaz, I feel like we all ought to just stop for a second and thank him, because without him, I imagine many of us probably wouldn't even be here today because we flat out wouldn't have survived childbirth. Disturbing for sure. All right. There were lots of issues when we think about giving birth to children. There are still issues. But back in the day, ladies would get their materials prepared to go to the hospital to deliver a baby. They would pack their bags. That stuff still happens, right? You get prepared, but they would also pack another bag. And this is, to me, really pretty disturbing. They'd get all their stuff ready so they could hatch out this kid, but they'd also prepare another bag that was full of their death clothes. Whatever it is that they wanted to be buried in, they would get that stuff together because the odds are not in her favor. Uh, Highly likely that you're not coming out of there. That's disturbing. Makes you wonder why even hatch out a kid in the first place, right? Disturbing. Puerperal fever or childbed fever were issues that many women ended up succumbing to. They would go in, the baby would be delivered, she would get a serious infection and would die. And oftentimes, mom dies, baby dies, right? Bad news, all right? So as we move in to Semmelweis's time, he was a physician, and he was working in a hospital. Lots of hospitals have a little bit of teaching stuff going on. There are lots of different potentials. All right. So he is at a teaching hospital. There are numerous medical students that are learning how to become doctors. All right. And in the process of doing this, it's like, your A&P lab. You've got donors or cadavers that you're working on, dissecting and understanding all sorts of features about the anatomy. These people are doing the same. All right. A woman comes in ready to deliver. They turn from the cadaver to her, deliver the baby back to the cadaver. All right. We'll wash our hands because why the hell would we need to? Right. We're men. Right. We're future doctors. Right. And in turn, you don't tell me what to do, right? Is ultimately a lot of the mentality, all right? He recognizes there's two different wings of this hospital. There's the wing with the doctors, we'll deliver your babies. There's the wing with the midwives, we'll deliver your babies, all right? Midwives, females, doctors, males. That's how it was pretty much back in the day, all right? So he recognized that the midwife wing, there was very, very low levels of childbed fever. But on the other side, it was skyrocketing. There were all sorts of issues. And he starts to wonder, what the heck's going on here? And he starts making some observations, as a good scientist might do. And he recognizes one feature that was different. The midwives washed their hands, right? Probably with some warm water because that would be nice, right? They washed their hands, and the doctors weren't doing that. So he starts trying to implement this idea. We ought to wash our hands. And the doctors were not happy about that. It took a couple of years truly to get this on board, because many people are like, you can't tell me what to do, right? I'm a stinking doctor, right? Or soon to be one, right? And in turn, they felt like they shouldn't have anybody telling them what to do, right? So there were a lot of people against him, bashing him, trying to destroy him because of what he was thinking, all right? But as soon as we truly started washing our hands, the doctor side of that hospital, their levels of infection mirrored that of the midwife side. All right. Some people, didn't matter which side, were still going to have issues. All right. But it was considerably lower. All right. Wash your stinking hands. Right. Take home message. All right. So I see Lister and all I think of is Listerine. All right. Little bit different, but still. So he started developing some procedures, not thinking hand washing, but still cleanliness. All right. As we start implementing all of these different ideas in medicine, we start seeing really big advances in people surviving. All right. So his ideas were thinking about care for wounds and equipment. We ought to clean that stuff. We ought to sterilize it, right? And he was using like some carboxylic acid to deal with cleansing and sanitizing equipment, all right? We already talked about Koch before with Koch's postulates, understanding specific agents that are culprits for disease. And both of these we think about huge, along with Semmelweis, huge importance when we think about infectious disease and a clinical situation. All right. You want to understand what the culprit is, and you truly want, you would hope, to ensure that you're not contributing something else into that patient. Done deal. All right. So here we can see, and this I believe is going with one of the case studies in the text, and we're not going to go through that stuff right now, but a radiograph with an individual with pneumonia and then normal lungs. So you can see the infiltrate within the lungs, indicative of that. There are numerous players that have the potential for this, and we will think more about that later. But for time, I'm not going to go into the case study today. All right. So this just gives us a bit of some historical stuff, putting it into perspective as far as timeline goes. So we can go back to ancient Greeks. They're the ones that had the thought of this foul smelling air, the miasma theory, that stuck with us for a super long time. All right. Malaria, a nasty parasitic infection, is named that for bad air, mal area, foul smelling air. How do you get this crazy high fever disease that we don't know what the heck it is? Obviously, you smelled some bad air. Technically, marshes and swamps, boggy areas, they're pretty stinky, but it's not the stinky air. It's all the mosquitoes that love that type of a habitat. That's truly where you're contracting malaria from, not the bad air, even though the swamps stink. All right. But it's made it stick, right? Malad, Idaho, Malad City, Idaho, because they want to make sure you know it's a city, right? A city of 2000. Malad, formality, early settlers putting down roots in that valley had a lot of issues. And that was traced back, most of it, the speculation, back to water, highly likely Giardia. Lots of people contracting that as they were first settling there. And there's the name. I'm like, that's awesome, right? Probably not if you're coming out of Malad. Anybody? No. Okay. You're like, don't say it. All right. Francastro, 1546, early version of germ theory, starting to think a little bit about this. Hooke, seeing the cells, century later. Leeuwenhoek, decade later, sees the single-celled, the wee little beasties, sees these with the microscopes he was building, all right? We jump forward 2 centuries and with Semmelweis and the hand washing. John Snow, we call him the father of epidemiology. He was able to figure out that this cholera toxin, this crazy stuff, another miasma idea, going clear back, we're still thinking that, right? He was able to show that the water was ultimately causing these issues. It was a couple of decades later before Vibrio cholerae was ever identified. The toxins were there. In epidemiology, typically you know the culprit and then you figure out that it's causing this epidemic or whatever. He didn't know the culprit, all right? Pretty cool, all right? We'll talk a bit more about John Snow a little bit later on with some thinking about disease and epidemiology. All right, 1862 is where Pasteur disproves, and remember I told you Tyndall, he's just a couple of years later, but we credit him just because Pasteur's famous, right? A lot more so than Tyndall, apparently, poor Tyndall. So that's this. And then Lister, Pasteur was actually a little bit before that too, 56 with fermentation stuff, all right? And his initial pasteurization wasn't milk, all right? We did that a little bit later. We're like, wait a minute, all right? The first stuff was frothy beverages, beer and wine. Wine's not frothy. Beer and wine have the potential to get contaminated with microbes, and then that stuff doesn't taste good anymore. You don't want to drink that. How can we prevent it? Pasteurize it, all right? And in turn, now you're good to go, all right? There's a lot of other thoughts with some of that stuff. I feel like, geez, should I just start spouting off beer stuff? Probably. It's a little early, but one other tidbit, India Pale Ales, the IPAs, they're called that, and it is an issue with transporting as the British were trying to colonize everywhere they could. As they were getting into India, they want their beer and they're shipping it over there. By the time it makes its way to India, that stuff is fouled and nasty and they dump it all out. How the hell can we get our beer here? Hops happen to have a lot of antimicrobial properties. So how can we help it? Throw a bunch of extra hops up into the mix, double hop it, all right? Get a bunch of hops in there, ship it over there, and now you got your beer. And that's where the IPA comes from. Extra hops, preserving it, all right? Delicious, all right? So there's other details about the British that I think are interesting. I'll save that for when we think some malaria, all right? How were they able to conquer places infested with these mosquitoes? Comes to some other beverages. All right. And then Koch with his postulates. So we showed this before, prokaryotic cell. This is kind of a generalization showing all of the potentials that we might see. And you'll see a handful of slides down, some ideas with some flagellates. But a flagellum, maybe a handful of flagella, maybe coming a tuft off of one end, could be all over, could be on two opposite poles. There's multiple potentials for how the flagella are on these bacteria, and we could differentiate them a little bit. We're not going to get bogged down with that in here, all right? But you'll see some details with it and some of the different features. Probably test worthy, betcha, all right? Some of them have the potential to form a pilus, a connection from one cell to the next. Typically, a plasmid inside of them is coding for the formation of that and the exchange of genetic material. We'll talk about some of that a little bit later, and I'll show you an image today of cells in the process of conjugating. So this imparts some extra advantages that that cell may not have if it lacks that plasmid, like resistance to antibiotics or abilities to feed on different things, or maybe even cause disease a little bit better. Gives them some sort of leg up, all right? Speckled inside of the cytoplasm are the ribosomes, free floating within here, and then the chromosome, circular. We say it's kind of central in the cell, and we call that region the nucleoid. Don't confuse that with the nucleus. It's not membrane bound. It's just an area of where it's at, all right? Some of them have some fimbriae, little hair-like projections that allow them to maybe stick in between cells. Capsule, sugary layers, also giving them a leg up to avoid immune detection. All sorts of potentials, all right? And we will get into some of the diversity with microbes in the next unit of material, and we'll see some details back with bacteria. The same with some eukaryotes in chapter 5. So as far as morphology goes, we've already seen a little of this previous chapter as we were thinking about some of the differences when we think about microbes, bacteria specifically. So you'll have the opportunity in the lab to do a bit of staining and see some of this. Typically, you're going to see cocci and bacilli, all right, or coccus and bacillus, singular, all right. That's pretty much the common players when we think about the microbes that we work with, one or the other, all right. So then some other potentials, kind of curved, shortened rods, more oval shaped, spirillum, spiral shaped, and then more tightly packed, more springy are the spirochetes, all right? So some of the morphology. There are potentials as far as their arrangements go, and this becomes more important when we're thinking about the cocci, because we can use some of the staining characteristics to really, you don't maybe necessarily know exactly who it is you're looking at, but you got a pretty good ballpark. And there's some different genera that truly can be kind of one way or the other based off of some of these characteristics. So they're all spherical. Sometimes they're just all separate individual cocci. That's not going to be super informative, all right? Other times you see them grouped up, and almost all of them grouped up. Diplococci, two of them together, and they replicate and they kind of stay stuck together, all right? It's all dependent upon planes of division as these cells are replicating that dictates some of these further morphologies that we might see. So a tetrad is a group of 4. Looks like just little 4 cells together. Sometimes they're stacked up. So now you got 8 of them. We don't have that up here, but a kind of cube of spheres. We call that sarcinia. All right. So 2 tetrads stacked on top of each other. The streptococci are chains of coccus together. Streptococcus pyogenes is the first one that comes into my mind, all right? And seeing it under a gram stain, big long chain of positive cocci. The staphylococcus, staph infections, Staph aureus, one of them. These cells are all clustered, all right? So all irregular. They divide in half, vertically, horizontally, and then diagonally. All sorts of different potentials. So you don't get just 4 side by side or 2 of them side by side. They're all over the place, like a cluster of grapes, all right? Irregular planes of division. And seeing chains or clusters clues you into streptococcus types of bacteria or staphylococcus types of bacteria. And some of the features of these are definitely different, all right? And then our rod-shaped, the bacillus. Sometimes we see them linked together, all right? That is not something that's as informative as the clusters and chains of the oxide, but might clue us in a little bit about some of it. But it's not something that I'm like, oh, these must be this type of organism because it's chained up. I see these and I immediately start thinking streptococcus, all right, or staphylococcus, respectively. So we need to think a bit about cells, and there's definitely differences between cells that have a cell wall versus those that don't, as far as some of the impacts. The cell wall helps to protect those cells, helps to give it some structure. And if it's just a simple membrane, they're more sensitive to shifts, osmotic shifts, and that can be detrimental to the cell. Food preservation can utilize some of that, all right? So as we look at this, we're looking at different tonicities, different concentrations from the inside versus the outside of the cell, all right? And the stuff, the solutes that are in the cell or dissolved in the fluid that the cells are in doesn't have to be the exact same materials, but the concentrations are what we're thinking about of these materials, all right? And that becomes important, all right? Just depends on what you got inside versus outside. If those solutes are the same on the outside versus the in, and as we see here in the first frame, it's basically balanced. And the arrows are showing the movement of water via osmosis, all right? The arrows are the same, in and out, all right? So there's water moving, but there's no net directional movement of water. We're just happy. Our cells want to be in an isotonic solution, all right? Hypertonic, there's a boatload more on the outside than the in, so those cells try to equilibrate their surroundings, and water goes out, trying to balance it. And in the process of doing that, cell membrane starts to shrivel up as we lose water. We call that crenation. If it's a red blood cell, that's bad news, all right? And these cells get super dehydrated, potentially dying, all right? The opposite is the hypotonic. Outside of the cell, very few solutes compared to the inside, and water in this case wants to go into those cells, trying to equilibrate it to the environment, all right? And in the process of doing that, instead of shriveling up, we swell up. And if you don't have a cell wall, the potential to rupture or lyse is there. And if it's a red blood cell, we call it hemolysis or homolysis, the lysis of red blood cells, all right? Bad news, all right? You don't want that happening. So cell walls, like I said, give it some structural integrity. There are, and this looks to me like some plant cells, but prokaryotic cells are what we're imagining. This looks multicellular to me. It looks like we're just looking at a bit of plant cell. Imagine, all right, that's the case. So here we can see, and this also makes me think plant cell because we got a central vacuole in there. Bacterial cells don't have this, all right? So water can move in and out of that. The vacuole basically acting like a little reservoir inside that can deal with the osmotic pressures. And in an isotonic, we're fine, all right? You can see the membrane, the cell wall. Imagine it's a bacteria without this, and it's just the cytoplasm that's giving and taking, all right? So in a hypertonic, the membrane starts to shrivel up. The crenation is happening, but the size of the bacteria, you don't recognize it being impacted, but it is getting damaged, all right? And water is going out, potentially fatal consequence. And the opposite over here in the hypotonic, water surging in, there are potentials for it to be detrimental to the cell, but for the most part, in these situations, the cell wall is really giving it some extra protection, keeping it from completely getting destroyed. All right. But there are definitely some potentials. If you think about like preserving stuff, making jam or jelly, when you do that, you take whatever fruit it is and you add a boatload of sugar. All right. As you do that, you're creating a very hypertonic situation. Most bacteria flat out can't tolerate that. They're not going to be growing there. All right. There are other potentials, things like fungus, molds may like it. They might be just fine. All right. So it could still end up spoiling, but the potential for bacteria greatly minimized. All right. So we'll touch a bit on that a little bit later. So this just showing the chromosome of our bacteria, circular, typically single, and the dashed line is trying to show this nucleoid region. So you can see little hints of the genetic material right here. So N's for our nucleic acids, and C, this is cytoplasm, the rest of the juice of the cell. And it's kind of grainy looking. My imagination is a lot of this stuff are ribosomes that we're seeing, giving it kind of the fuzz within there. So bacteria, everybody's got ribosomes, protein workbenches where messenger RNA gets sandwiched around it, and then transfer RNAs start coming into the mix. We'll go through a little bit of that later on. But they're slightly different. Size-wise, prokaryotic ribosomes are smaller than eukaryotic ribosomes, all right? Function, the same, all right? And we mentioned before, recognizing some differences in this, truly the small subunit, the DNA coding for the production of the small subunit has some variation between bacteria and the archaea. Obviously, there's variation between prokaryotes and eukaryotes. They're bigger, but the variation between those two is what stimulated the separation into two domains, bacteria and archaea, and the third domain, eukaryotes, which was initially a kingdom by itself. All right. So there are potentials for bacteria or prokaryotes to have some inclusions. These aren't the same as we think about in a eukaryotic cell with membrane-bound vesicles and different roles. These are different, but there's still some sort of substance inside of the cell, or potentially maybe going to the outside. It just depends. All right. So lots of different things, from lipids to some pigments. There's sulfur granules and magnetosomes, like little magnets. All right. And they use these in an area kind of depleted in oxygen for some scavenging of that. We'll think the littlest bit about some of those ideas later on. All right. So we've talked a bit about some bacteria having potentials to form spores. I mentioned that with Tyndall and his process working with the vegetable broth or whatever it was, all right, that had some bacteria on it that could form spores. And I mentioned Bacillus and Clostridium as the two main spore-forming genera of bacteria. Technically, there are a few others, but as far as like disease stuff goes, they're the ones that we really think about. Not all spore formers are disease causing, all right? This one, Carboxydothermus hydrogenoformans, is another spore former and something that's non-pathogenic, all right? So this is the process of sporulation up above. Here we've got a spore that has formed, and then through some spore staining techniques, you can also see the bacteria in pink, and the darker bits inside of some of these bacteria, these are the spores. potentially give it another day and do this stain, and you're going to see a lot less of the vegetative cells and just spores. All right. Kind of just depends on the age of that culture as far as how is it going to appear. All right. So what happens in this process is, like that cell is going to divide, going through the process of binary fission, but we don't. All right. We do something different. The cell is receiving some chemical cues from its environment that stimulates this process, basically saying something's not right. Temperatures, concentrations of stuff, or the lack thereof, whatever it is, makes it sense. This is an inhospitable environment. I'm flat out going to die, right? So it replicates its DNA, but instead of separating and creating two new cells, as it replicates the DNA, it takes part of that, one copy of it, and starts to encase it in material that's going to be the protective layer around this nucleic acid, allowing it to persist through almost any sort of adverse environment, except for cooking it up at autoclave temperatures. All right. They could persist in the environment indefinitely until the conditions become favorable again. All right. They could live, if you will, they could live on decades or centuries later if it finally becomes a suitable sort of habitat. There's all sorts of things that I've, seeing in the headlines talking about, climate change, permafrost, all sorts of things melting. What about some of these organisms frozen away that now are thawing out? Are they harboring some sort of crazy, weird infectious disease? Is there something in there? Could they be holding inside of them some sort of virus or weird infectious agent that we haven't seen for millennia, and now all of a sudden here it is, right? Maybe a spore-forming organism could be up in there, and it was just frozen away, hanging out. Thaws out, conditions become favorable, and it starts growing. Who knows, right? I think it's impressive, all right? So it packages it up. It starts putting more and more material around it, protecting it, all right? And then it gets to the point that this vegetative cell ruptures, releasing that spore, and these pink guys would go away. All that's left would be the kind of bluish one. We use malachite green. This doesn't look green to me, but malachite green is a spore stain, and they would look like little green kind of ovals. Some of the spores make the cells swell. Some of them form at one end, the other, the middle. It truly kind of depends. And we can use some of the spore staining and how they do that to differentiate a few of them. All right. So that's spore formation. All right. So here, more membrane. We saw this stuff before, and their plasma membrane has all sorts of materials. We will move in and see cell wall here in a second. All right. But this allows for nutrients to come in, other substances to go out through the membrane. All right. And that becomes important. Most of the stuff when we're thinking about our microbes is just simple diffusion, concentration gradients being greater on one side of the membrane versus the other. And same sort of an idea, trying to equilibrate like that isotonic solution. Molecules have the potential to move through the membrane. In the case of diffusion, it's the solutes, sugars, or whatever it might be. It's the solutes that are moving, all right? When we think osmosis, it's the opposite. It's water that's moving. Those images that we showed before, it was water, not solutes moving, all right? This is solutes, all right? From high to low concentration. And then it equilibrates. Technically, we could show some arrows. They're all trying to go in this way, but once we get equilibrated, both directions. There's the movement. It's no net directional movement, but molecules are still moving around, all right? So sometimes it just can't readily move through there. There are protein channels that can allow some things in or out, maybe both, all right? It's not just like an open window that lets anything in. There are specifics based off of the respective molecule. Some of these proteins allow it in. Some may need a different protein channel, and then they can get in, all right? Some of them can open and close. Some of them stay open. Some of them require energy for this process to happen. And then it's not facilitated diffusion, then it's active transport, all right? But it's still a protein channel, and it's energy that's pushing things against concentration gradients instead of moving down them, all right? Active transport goes against the concentration gradient, establishing more stuff on one side of the membrane than the other. And oftentimes that stimulates some sort of charge imbalance. We could do something with that, all right? Maybe we could make energy, all right? So this is the active transport, sodium potassium pump. As we see this, inside down below, cytoplasm, outside up above, extracellular fluid. And this cell, sodiums, as we're opened up to the inside, sodiums can link up on the inside. And when we get them, 3 of them link up on the inside. ATP links up to the protein channel. Once it donates its phosphate to this channel, active transport, it's a pump that's using energy, right? We're using some energy. It causes the protein to change its shape. Closed to the outside now becomes open to the outside, and it shoves those sodiums out. While we're still opened up, potassiums hitch up into this protein channel, and then once they're locked up in there, we change our shape and shove the potassiums in. Sodium's out, potassium's in this pumping fashion. And you can see the concentrations of sodium accumulating out, potassium's accumulating in, and a charge imbalance across the membrane, all right? Muscle cells rely on these types of activity all of the time, all right? So we've talked about gram positives and gram negatives. We talked about gram staining and how we can show variations in the cell wall. This is some of the detail, although it just looks like some stick figures. This is some detail about how these cell walls of different bacteria appear. So the gram positives have a super thick layer of peptidoglycan, sugars and proteins and sugar. Peptid is the protein and glycan, the sugar, all right? So, just layered on top of one another and stitched together, all right? Versus the gram negatives, single layer, all right? I think of it like a bed with a single sheet, or a bed in the winter time. You probably want a couple of blankets up on there, right? Multiple layers, keeping you nice and toasty, all right? Different, all right? So the gram positives, thick layers, and gram negative, single layer. The gram negatives also have an outer membrane. We'll get to that in just one second. So there's a couple of protein sugar combos, NAG and NAM, N-acetylglucosamine and N-acetylmuramic acid. These guys link together to form your sheet. And then there are some other proteins that do the stitching together. So it's not just one sheet, it's 15 sheets all stacked up to make you a super nice thick comforter, all right? So spanning and stitching together, all right? Over here, it's just the NAG and NAM, and then linking those together with these tetrapeptides from one group to the next group, making the threads to make your sheet. And then the pentapeptide is what's making them multiple layers. We lack that. It's only a single layer, all right? And gram staining ultimately tells us this. Does the stain get stuck in the super thick blanket of cell wall, or is it easily pulled out of it? And then we know some details about those microbes. So positives versus negatives. Here's the outer membrane, that lipopolysaccharide layer, and it is easily degraded as the alcohol touches it, and it pulls that color out of this thin peptidoglycan layer. All right. There's other stuff. Positives have this teichoic acid that basically is like a tether from the plasma membrane spanning through this peptidoglycan, basically keeping your blanket stuck to your bed, right? You don't want that sucker moving, right? That's what the teichoic acid is kind of helping it to do. The negatives lack that, all right? There are some other potentials as we see tethering from the outer to the peptidoglycan layer, and a bit of space in between the membrane and our peptidoglycan layer right here. Technically with this, I mean, there's a little bit of space, but it's cell wall peptidoglycan butting right up very close to the plasma membrane. Still a little bit of space, all right? We will think some metabolism, mitochondria, we'll talk about with our eukaryotes, but they were once prokaryotes, all right? And in bacteria, the proteins that are needed for the electron transport chain are studded on the membrane. In bacteria, they're studded on this plasma membrane, and they push hydrogen ions outside, establishing a concentration gradient, either right here into this periplasmic space or in between the peptidoglycan and plasma membrane, establishing a gradient, and then ATP synthase recharges those molecules. So there are some other potentials, stuff like tuberculosis, that are acid fast. They've got a waxy shell called mycolic acid. It's not the same as the lipopolysaccharide, but it gives it some extra protection, all right? Water doesn't really like those lipids, right? And that wax layer makes it so that these cells don't pick up a Gram stain, all right? Unless they're old and starting to get a little bit degraded, the mycolic acid might break down. Then they may appear as Gram positives, all right? But typically they don't show up in a Gram stain, all right? So they are Gram positive bacteria. They do have a very thick cell wall, but we don't really recognize them as Gram positives because the Gram stain doesn't work with them because of the mycolic acid, all right? But they are Gram positives, all right? They're just cloaked from that Gram stain. And we'll touch on TB and some others as we progress along. So in our Gram negatives, there are some potential issues, and some of those issues can be pointed directly at the outer membrane of the bacteria. There's a few things that they harbor that can stimulate an ill effect in the host. And it also sets the stage when we think about the potential to treat with antibiotics, dependent upon who we're talking about and where that infection is. Sometimes killing Gram-negative bacteria with antibiotics sets the stage for the symptoms to become more pronounced because you're releasing more of these toxins, all right? So O antigens can stimulate an immune response, and then the lipid A down below also has the potential to stimulate some like virulence factors enhancing the disease, all right? So we'll touch on a little of that later on. This image shows some of the capsule potential with our bacteria, a halo around them. It's another potential for these microbes to be a little more infectious. If our body doesn't see the bacteria, it only sees the sugar. Our body says sweet sugar, right? Doesn't see what's hiding underneath. And it allows those bacteria the potential to replicate, gain hold, and truly establish disease, amongst other things, all right? So bacteria don't have sex, but kind of. All right. That's what we got going on right here. All right. It makes me feel like, man, maybe we should have blurred this image out, right? So we've got two bacteria and there are pili making some connections between them. All right. Looks like another pilus right here. All right. Other little projections, fimbriae, little hair-like projections. All right. But the pili that we see here are making connections with the bacteria. All right. We got a couple of them going on here. All right. Some plasmids code for the production of this. Some don't. Some just have whatever advantage. But in this case, a pilus forms, that plasmid replicates, and it gets piped over to the bacteria that doesn't have it. Now both of them have it. This is how stuff like antibiotic resistance can show up in a class of bacteria that never had it before. They can share it with somebody that's nowhere even near them. All right. I mean, close to them, but genetically nowhere near them. All right. So it imparts some differences. It's not recombination in the chromosomes. It's not like haploid to diploid, and here you are. All right. But it does give them some different genetic information. There are also potentials for these plasmids to maybe take with them part of the host chromosome, and then that makes it incorporate into the new recipient's chromosome instead of hanging out by itself. We'll touch on a little of that stuff later with some genetic exchange. All right. So the flagella, I'm running out of time. The flagella with these similar, it's a little bit different, negative versus positive, just to do with the cell wall material, the outer membrane, but deep down it's all basically the same. Stuck into the plasma membrane of the bacteria, tethered in there, is basically the motor for the flagellum. And these spin like the propeller on a boat. All right. In eukaryotic cells, it's microtubules cross-linked together, and they shift back and forth like this on themselves, 9-2 arrangement. When they're doing this, you get a whip-like motion, like a sperm cell, a eukaryotic flagellated cell, all right, whipping back and forth. Bacteria spin like a propeller, all right? They do things really kind of differently. They can sense stuff and move chemotactically toward it, and they move by runs and tumbles. That's the next image or two images where we'll see how they do that. And it's not a directed movement. It's a little sporadic. So a single flagellum, we call that monotrichous, one flagellum poking off of one end. Amphitrichous, opposite poles. Lopotrichous, a tuft of flagella coming off of one end. I think that's a test question. And paratrichous, this is like little hairs all over the place, everywhere, all right? When you are in drive, when you're trying to go forward, they are all turning together, and I believe it's counterclockwise. We move forward. When we want to go in reverse, we flip it in the opposite direction. If you're a single flagellum, whatever. But the others, now they just behave differently. That gives you the tumble because none of them are working together and it's all sporadic. All right. So tumbling, everybody doing their own thing. And then running, we all function together, spinning the same and propelling us forward. So you don't get there just by turning like this. You got to go forward. You got to tumble. Maybe I tumbled and now I'm pointing this way. I got to tumble again. Now I'm tumbling back here. Finally, I get there. All right. But if there's some substance that I can smell, that I can sense in that environment, I'm going to do more runs and tumbles than if I'm just hanging out in this isotonic stuff and there's nothing for me. All right. So that's the idea with it. All right. Real quickly. All right. Are eukaryotes, all sorts of features within here, membrane-bound organelles, lots of structures in here that make them considerably more complex than the prokaryotes. All right. A lot of diversity, some flagellated, some not, some intracellular, some not, all right? Just depends on who we're talking about. I feel like you should have a pretty good handle of structures inside of a cell. What's a lysosome? What's Golgi doing? What's up with the nucleus? All right. Those features are really just a few images in here. The rough endoplasmic reticula butting up against the nucleus is where we think about a lot of proteins that are going to function as inside of a vesicle or maybe be transported to the surface. Other things staying in the cytoplasm interact with free-floating ribosomes, but most of them go this way, all right? Golgi apparatus, along with the smooth endoplasmic reticula, snag molecules that have either been proteins generated through the rough endoplasmic reticula, other things coming in membrane vesicles from the smooth endoplasmic reticula, like maybe some lipid, also providing membrane material. These show up at Golgi, packaging it together and blubbing it off with all of its stuff. Lysosomes are full of boatloads of proteins, digestive enzymes, each one of them made by its specific messenger RNA, shipped out of the rough ER, making its way into Golgi, packaged up, blebbed off out of there. Now you got these lysosomes with 50 different enzymes in them, all right, as it packages and sorts, all right? And that really deep down is about it. Some of the tubules, like I said, microtubules allowing for movement, cell cytoskeleton, shaped like that is the idea. All right. That's, although there's a bit more in here, I think sufficient. A quick little review of that stuff without me spouting it off. I think you will be just fine. You're not going to have boatloads of questions about that stuff in the first place. The test will open not immediately because I didn't make it available, but I'm going to get back to my office. It should be up 30 minutes or so. It'll be up there. Questions, concerns? Farewell. Have a good weekend.